Angenmed Therapeutics, pioneering neoantigen-targeted therapeutics for precision medicine to eradicate cancer. Immune cells, especially dendritic cells as trainers and T cells as effectors, play a crucial role in the elimination of cancer cells. Since neoantigens are specific to tumor cells, they can be recognized as foreign, making them promising targets for cancer immunotherapy. Additional, neoantigens are tumor-specific. When cancer cells are destroyed and tumor cell debris is released, dendritic cells can uptake the tumor cell debris and then present the tumor neoantigens to T cells to activate T cells. After T cell receptors recognize neoantigens presented by HLA on cancer cells, the activated T cells secrete cytotoxic proteins to lyse the cancer cells. This specificity helps in developing personalized cancer vaccines and therapies that aim to stimulate the patient's immune system to more effectively target and attack the tumor cells. Introducing our proprietary AGT NeoSync platform, AGT NeoSync. The patient's tumor samples are collected for DNA sequencing and bioinformatic analysis to identify the somatic mutations and to generate individual neoantigens AGT personalized neo. Then, the patient's peripheral blood mononuclear cells are collected through leukapheresis or blood drawn. Angenmed Therapeutics will process the monocytes and T cells to create personalized dendritic cell vaccines and T cell products tailored to present and recognize the patient-specific neoantigens. These dendritic cell vaccines and T cell products will be infused back to the patient to target and kill the cancer cells. Neoprime DCs. Dendritic cells presenting neoantigens can work as trainers to educate T cells to recognize tumor cells. NeoBoost T cells. Neoantigen-specific T cells are expanded ex vivo. The infused NeoBoost T cells can recognize and destroy cancer cells specifically. Angenmed Therapeutics has already developed 23 panels targeting the common neoantigens of different types of solid tumors. Neoantigen AGT Core Neo. For example, KRAS mutation is present in several solid tumor types, including pancreatic cancer, colorectal cancer, and lung cancer. We have expanded strong T-cell immune responses to achieve high neoantigen coverage of KRAS mutation, even with only one mutated site. These specific T cells can recognize and attach to the tumor spheroids with KRAS mutations. Then, these T cells will release cytotoxic proteins to induce morphological alteration and cell death signals of tumor cells. Moreover, compared to other cancer vaccines, we have developed the Neoprime DC vaccine, which introduces over 200 public or personalized neoantigens ex vivo, transforming them into professional antigen-presenting cells. These antigen-presenting cells can precisely present the introduced neoantigens. Angenmed Therapeutics' two-step neoantigen targeting treatment strategy includes AGT Core Neo, a ready-to-use neoantigen panel from 23 solid cancer types, and AGT Personalized Neo, a customized personal neoantigen panel. Our AGT NeoSync platform can present all epitopes of cancer neoantigens to T cells. T cell receptors on the trained T cells can distinguish only one amino acid residue difference on neoantigen epitope from the wild-type peptide in normal tissue. This precise recognition enables strong cytotoxic T cell immune responses to effectively kill tumor cells. Compared to other immunotherapy platforms, our AGT NeoSync platform has abundance of neoantigens, includes targeting driver genes, produces cells with long immunity potency, and has higher yield, stability, and functionality of cell products. AGT NeoSync platform offers potential best-in-class cancer immunotherapy, including DC vaccines and T-cell products that target comprehensive tumor neoantigens, enhance specificity and functions of CD8 and CD4 T-cell products, and high efficiency in our manufacturing processes. We will be launching clinical trials of DC vaccines and adoptive T-cell therapies towards several solid cancer types, including Lynch syndrome, sporadic CRC, urothelial cancer, renal cell carcinoma, and ovarian clear cell carcinoma. These multi-center clinical trials will be conducted to fulfill the unmet needs of high-risk populations in the United States, Japan, Taiwan, Korea, and Singapore. We also offer broad options of clinical therapies for cancer prevention and treatment services in Japan under Regenerative Medicine Class 3. Angenmed Therapeutics is a global cell therapy company. Our headquarters and R&D center are located in Taiwan, driving innovation and ensuring continuous pipeline improvement, clinical trial conduction, and product development. The manufacturing plant in Taiwan is responsible for AGT Core Neo and AGT Personalized Neo production. 4 state-of-the-art manufacturing plants will be located in Taiwan, Japan, UAE, and SEA. These facilities are dedicated to generating Neo-Prime DC vaccine and Neo-Boost T cells with the highest quality standards for clinical trials and clinical service.